Question number 1. In a DNA molecule, deoxyribonucleotides are joined into a polymer by the covalent linkage of phosphate group. The covalent link term as hydrogen bond, glycosylic bond, phosphodiester bond, covalent linkage. And correct answer is option C, phosphodiester bond. Question number 2. The structure of RNA differs from that of DNA as RNA contains the sugar ribose instead of deoxyribose, uracil instead of thymine. It contains cytosine or D, A and B. Correct answer is option D, A and B. RNA contain ribose sugar instead of deoxyribose, uracil instead of thymine. Question number 3. Two separate chains of DNA are wound around each other, each following a helical coiling path resulting in single-strand coiling, a right-handed double helix, alpha helix primary structure. Correct answer is option B, a right-handed double helix. Question number 4. Replication of bacterial chromosome normally start at a fixed point called replication for recognition site ORV ter. Correct answer is option C, ORV. Question number 5. Which of the following is true for the mechanism of excision repair? Endonuclease cleavage removes a portion of damaged strand. The gap is filled in by DNA polymerase 1. The sugar phosphate backbone is resealed by DNA ligase. All of above correct. Correct answer is option D. All of above are correct. Question number 6. The term cistron refers to region in tRNA molecule, codon, region of the DNA that codes for single polypeptide chain, ribosomal protein. Correct answer is option C, a region of the DNA that codes for single polypeptide chain. Question number 7. Bacterial ribosome typically consist of two subunits. The larger 50S subunit consists of single RNA molecule 60S and 21 polypeptides. Two RNA molecules 30S and 50S plus 31 different polypeptides. Single RNA molecule 80S and 21 polypeptides. Two RNA molecules 23S and 5S plus 31 different polypeptides. Correct answer is option D. Two RNA molecules 23S and 5S plus 31 different polypeptides. Question number 8. Bacterial ribosome typically consist of two subunits. The smaller one 30S contain single RNA molecule 60S and 21 polypeptides. Two RNA molecules 30S and 50S plus 31 different polypeptides. Single RNA molecule 80S and 21 polypeptides. Two RNA molecules 23S and 5S plus 31 different polypeptides. Answer is option A. 30S subunit contains single RNA molecules 60S and 21 polypeptides. Question number 9. Which of the following is relevant for ribosomal attachment of specific sequence in bacteria? The ribosome binding site, the signed organo sequence. This sequence is partially complementary to the 3' end of the 16S rRNA. All of above is sufficient. Correct answer is option D. All above is sufficient. All are the true answer statement. Question number 10. Which of the following is true for using genome Southern blotting to identify the sequence, to identify the number of sequence, to identify DNA fragments, to identify RNA sequence? Correct answer is option B, to identify the number of sequence. Question 11. The stringent response is triggered by which leads to the presence of uncharged tRNA occupying the A site of the ribosome. mRNA, Lysine, amino acid starvation, water. Answer is option C, amino acid starvation. Question number 12. The base composition GC percentage of bacterial DNA varies widely from one species to another, but closely related organisms tend to have similar DNA base composition over a range of 20 to 80%, over the range of 10 to 20%, 5%, 10%. Correct answer is option A, over a range of 20 to 80%. Question number 13. The genera Staphylococcus and Micrococcus are morphologically similar gram-positive cocci. However, Micrococcus has high GC content, about 70%, whereas Staphylococcus DNA has low portion of GC content, 30 to 39%. Staphylococcus has high GC content, whereas Micrococcus DNA has low portion of GC. Micrococcus has high GC content, about 30%, whereas Staphylococcus DNA has low portion of G plus C, 10%. Staphylococcus has high GC content, about 70% GC, where Micrococcus DNA has a low portion of GC, about 30. Our answer is option A. Question number 14. Bacteriophage T4 is a virulent bacteriophage of E. coli which contains a linear DNA molecule about 165 kb, a circular DNA molecule about 50 kb, a linear DNA molecule about 40 kb. All of the above are false. Correct answer is option A, a linear DNA molecule about 165 kb. Question number 50. Which of the following is true for disruption of secondary and ternary structure of protein by heat or extreme of pH? Denaturation of the protein, formation of random coil formation of ternary structure. A and B correct. Answer is option D, A and B correct. Denaturation of protein and formation of random coil conformation. Question number 60. In nucleic acid, the bases are covalently attached to the first position of pentose sugar ring to form a nucleotide, DNA single strand, nucleoside, glycosidic bond. Answer is option C, nucleoside. Question number 70. The bond between base and the sugar in DNA refers to hydrogen bond, glycosidic bond, phosphodiester bond, covalent linkage. Answer is option B, glycosidic bond. Question number 80. Which of the following is correct for A form of DNA structure? A. Wider than B form. More compressed structure in which base pair are tied with respect to the helix axis and actually lie off the axis. The helical repeat of A form is around 11 base pair per turn. All of the above are correct. Answer is option D. All of above are correct. 90. In strong acid and at elevated temperature, for example, perchloric acid HClO4 at more than 100 degrees Celsius, nucleic acid tends to break the some hydrogen bond, hydrolyze completely to their constituent, denaturation of some nucleotides, denaturation of some nitrogen bases. Answer is option B, hydrolyze completely to their constituent. Question number 20. The effect of alkali to change the DNA structure, deoxyribose sugar, phosphodiester bond, tautomeric state, GC content. Answer is option C, tautomeric shift.